Welcome along, ladies and gentlemen, to your weekly SETI seminar series. Today we're joined by Jan Liphardt, who's come to us today from UC Berkeley. Jan did a BA at Reed College in Portland, Oregon, and he got his PhD from Cambridge University in the UK. Then he came back here to the Bay Area for his postdocs at UC Berkeley, where he became faculty in 2004. He's currently the director and PI in charge of the UC Berkeley Physical Sciences and Oncology Center. And his science research focuses on techniques to study DNA unfolding, bending and cleavage. And today he's going to talk to us about the evolving intersection between physics and biology. So if you'll join me in welcoming Jan.
Okay, it's a pleasure to be here. Just to give you a little bit more background, I'm also with the Physical Biosciences Division at LB&L, and of course I'm part of the physics department at UC Berkeley. And since focusing on single molecules, this was quite a while back, we really moved on a little bit as a lab to study primarily cells and collections of cells, things like tissues. But what I want to do today is actually something that always surprised me that we don't do more of, which is to give talks about things we don't understand. It seems like most science talks are about things we think we understand, and so what I'll try to do today is I'll enumerate things that we're pretty certain that we in fact do not understand. And so what my general focus will be on things that we would really like to understand, and I'll try to illustrate what some of the implications are in biology. So let's take a big step back, at least 100 years, and what was the case in those days is that biology involved an array of subjects. So typically someone studying biology, turn of last century, they were also interested in chemistry and geometry and physics. And a classic problem in those days, 1905, 1910, 1920, one of the things that fascinated people typically were patterns. And so they looked at snowflakes, they looked at seashells, they looked at honeycomb patterns, they looked at wings of flies, of Drosophila, they looked at how bones are shaped. And as they studied these biological objects, these patterns, these regularities, they were interested to think about what those patterns meant for how mechanical forces, how cell and tissue mechanics played a role in biology. So 100 years ago, 110 years ago, we have this mixture of chemistry, geometry, physics, and biology. Here's some more contemporary examples of that. So here's a beautiful image of a snowflake. This is by a colleague in the Caltech physics department. And probably for everyone in this room, since you were a little kid, and the first time you looked at a snowflake under the microscope, I'm sure you were very curious about how it could be that every single snowflake was different, and you could generate these absolutely beautiful symmetric patterns with something as simple as cooling water. The other thing, of course, if you live in the Bay Area, we have excellent access to yuppie supermarkets. And so you'll be able to buy various kinds of vegetables and fruits that also excite mathematicians, You can do a whole lot of mathematics by studying Romanesco broccoli, various series, and a wide variety of expressions. And what has happened since then? So we have this track of mixing patterns, biology, physics, geometry. And at the same time, probably since the 1950s, 1960s, we have this real explosion of molecular biology and biochemistry. And so the big ideas here are we're really interested in the gene. And one of the prevailing notions was that a lot of the big mysteries in biology are about reading the entire genome. So the prevailing idea was, we know what a few of these genes look like, but what we really need to understand, what we really need to do to read the entire blueprint of life is we need to start sequencing entire genomes. And one of the prevailing notions was that once we've done that, once we've sequenced entire genomes, then we will be able to sit down with a very large cup of coffee, and we will be able to read the genome and figure out many of the secrets of life. And of course, the human genome was sequenced in 2004. We had this notion that there are typically simple relationships between a small number of proteins and certain diseases. Protein X causes disease Y, or a mutation X causes disease Y. One of the other notions of this era of biology is one of the interests certainly is an interest in this relationship between protein structure and protein function, and also beginning to create networks, beginning to map out interaction networks among proteins. And so a classic thing to do last decade, and certainly also this decade, is to map out interaction networks. Each one of these dots is one protein, and the name of the game is to connect proteins with arrows denoting a physical interaction. And so just imagine all the people in this room. If I were able to somehow track all your conversations, I would learn over time that certain pairs of people tend to talk more frequently with one another, and that would allow me to construct this kind of interaction network. And so, of course, biological networks in cells or tissue typically don't have 10 or 100 interacting components, but they have tens of thousands or millions of interactions. And of course, you can keep making these diagrams ever more intricate. And this is a quick schematic of what we now know of the interaction network, the signaling network that's important in cancer. And so each one of these arrows has been laboriously established, typically by wet lab biochemistry experiments. Against many of these arrows, we now have drugs that allow us to modulate interactions between any of these pairs. And what's shown here is about 10% of what we now know about signaling in cells. And so the actual network of these interactions is considerably more complicated. So now the genome has been sequenced, right? That happened in 2004. We have crystal structures of most of the key proteins. We know structures and functions for quite a few proteins individually. And we have a general sense of the major themes of these interaction networks. And so at this point in 2010, we can ask a simple question. How much of biology do we actually understand? And is biology over? Are we basically done with biology? And if we want to answer that kind of question, what kind of grading scheme do we use? How do we know when biology will be over? When are we done? And the most useful thing strikes me in this context is of course, Richard Feynman's blackboard the day he died. And let's see, so know how to solve every problem that has been solved. That's certainly a very useful thing. There are some solved problems, some problems that have been since solved. But there is his general dictum, what I cannot create, I do not understand. And So let us use this scorecard to get a general sense of where we are in this field of biology. So if that's our metric of how we're doing, so the question arises, how much of the biological world around us can we recreate from first principles? So the question is, if I gave you a piece of paper and a pen, and I told you to build me some biological device or object that performed a certain function, the question arises, how successful would you be typically? And the answer is you would be, on average, extremely unsuccessful. And so it turns out, at least this is my personal opinion, that we're still only taking extremely small baby steps when it comes to being able to design and create things following Feynman's dictum. And so the question arises, why is that? Are we simply not very good at that? Is this a matter of scale? Do we need larger universities? Do we need more robots? Do we need larger computers? Do we need more money? Are there critical infrastructure needs? Are scientists, are biologists not very dedicated? Do they have a lack of interest? And the answer to all those things is no. And so what I think instead is going on is that there's a significant lack of knowledge of fundamental biological organizing principles and mechanisms. And so what you typically see in biology, and I am a biologist primarily, and so I'm criticizing, this is a lot of self-criticism on my part. And the other aspect to this is that if you're in physics, at least in particle physics and things like that, there is a standard model for what you're doing. And so you have this overarching framework for how to design experiments and so forth. And biology, to this day, really doesn't have that yet. And so life would be a lot easier if there existed this kind of standard model, but we really don't have that. What we're very good at in biology is we're very good at characterizing these individual pieces. And so this is a container filled with Lego blocks. And I have all these individual pieces. I can count them. I can ask what the distribution of colors is and so forth. But of course, what we really want to know is how biological systems function, like this Lego engine, and parts are going to turn with respect to other parts and so forth. And of course, what's noteworthy in biology is that not only do these biological systems have interesting functions, but of course they self-assemble themselves and they repair themselves and all these other things that, of course, a simple object like this made of Lego blocks can't do. And so what I'll do at this point is, that was a little bit of a general introduction. What I'll do at this point is focus on three very specific problems that I think it would be very useful to actually figure out. So problem number one is relating genes to cell types. And I'll clarify what I mean by that in a moment. So how many genes do you guys have, right? We used to think something like 100,000, of course, because humans are, quote unquote, more complex than fruit flies. It turns out that we have, roughly speaking, the same number of genes as many other animals. So we're talking about 23,000. Only a small portion of our genome actually codes for proteins. The 99 or the 98.5% that doesn't code for proteins was once called junk. And as we now know, most of what that is doing is actually playing various regulatory roles. And so what you have is a rather small number, 23,000 proteins, but you have an exquisitely complicated set of regulatory mechanisms. And so a lot of the complexity in our genome does not reside in the proteins we encode, but the regulation of those proteins. And so if you look at proteins, in fact, this is a picture of a cell in fluorescence microscopy. The scale bar here is maybe 50 by 50 microns. And so a human hair is maybe as thick as the slide. And so you're talking about half of a thickness of one of your hairs. And what you're seeing here is in red, the cell membrane, blue, the nucleus, and green, the cytoskeleton. And so every cell has a very well-defined cytoskeleton that has a number of sort of mechanical roles and also regulatory roles. And if you ask for these 23,000 proteins, where are they? How many are there? what you'll find is that protein levels and also spatial organization is exquisitely well regulated. And so you have an enormous number of regulatory possibilities. You have various pre- and post-translational regulatory mechanisms. You can control nuclear export. You can alternatively splice proteins. You can do all these other things. And so every one of these proteins is subject to exquisite regulation, concentration, activity, and location. And this can now get pretty complicated pretty quickly. And so there's a famous lecture by Richard Feynman where he is discussing gravity. And an old lady, and many of you will know the story, an old lady raises her hand and says, Dr. Feynman, everything you said about gravity is wrong. The Earth is actually sitting on the back of a turtle. And Richard Feynman says, but what is that turtle sitting on? The old lady says, it's turtles all the way down. And so one of the issues with all these regulatory possibilities is really this turtles all the way down problem. And I'll illustrate that in a moment. So let's just think about one of those many regulatory mechanisms, which is alternative splicing. And so for each one of those 23,000 proteins, that protein is composed of a few exons. And so here's one gene, and the protein is encoded by 4 exons: yellow, red, green, blue. And so what the cell can do is it can take the yellow, the red, the yellow, the green, the yellow, the blue, the red, the yellow, the red, the green, the blue, the green, the red, and so forth. And so you can imagine each of those 23,000 genes, I can now create many, many different proteins from that. In fact, sort of 3 or 4 factorial from that one gene. And so now just imagine for a moment, so some genes have as many as 12 isoforms. So 12 different proteins being encoded by one gene. And just imagine what this means for a cell. So for one cell, you can make an absolutely staggering number of distinct sets of proteins per cell. And this is an enormously, enormously large number. And yet, look in this room. So if I take my glasses off, you basically all look very, very similar. You all have 2 eyes on average. You have 10 fingers on average, and so forth. And so you can begin to see where some of the basic issues arise in sorting this out. And so if I look at you more closely, what all of you have is on order of 210 or 300 or 400, we can argue about that, a few hundred distinct, discrete cell types, things like neurons, fibroblasts, and so forth. And so the complexity arises in, on a microscopic level, we have an absolutely astronomically large number of distinct microscopical sort of states of a cell, if you want. Now, on the macroscopic level, we have a few hundred well-defined cell types. And so what I'm drawing here is some sort of phase or type space or something, where the color denotes maybe this could be a neuron, this could be a fibroblast, and this could be something else. And what I think must be true, and this is to some degree for a statistical physicist, this is completely not surprising, it must somehow be that the system is highly degenerate. It must be that microscopically, cells from one cell to the next, to the next, to the next, can vary quite dramatically, and still, they're all neurons. So the system, in some sense, must be highly degenerate. Many different combinations of pathways, genes, microstates, and so forth must all lead to the same general cell type. And so at this point, you might say, okay, this is beginning to make sense to me. Because if I care about how water freezes, it's well appreciated by any undergraduate that I do not have to track the individual molecular trajectories of every single water molecule. Rather, all I care about is I care about the temperature and 1 or 2 other parameters, and then, as usual, bulk thermodynamics kicks in, and I can make statements about entropies and phase transitions and so forth. And then I have a relatively small number of distinct states of water, ice, liquid, and so forth. But now there's another wrinkle on the subject. It turns out that it is possible to switch a particular type of cell from one type into another. And here's one recent paper on the subject. The title is Direct Conversion of Fibroblasts to Functional Neurons by Defined Factors. And so what you can do is you can take a fibroblast, something from your skin, and you can apply a very small number of signals, and you can turn that into a functional neuron with electrical activity and all the other stuff. And so what's really interesting about this is that, microscopically speaking, we have the system which has an absolutely enormous number of possible distinct microscopic states. We also have good reason to believe that the system is highly degenerate. You only get a few hundred distinct cell types in your body. By the way, how many cells are in your body? About 10 trillion, right? Just order of magnitude. And yet, it is possible to switch cell types from one type to another by a small number of distinct factors. And so that's telling you that in this whole mix of things, there are a few proteins whose concentration or localization or some other aspect matters a lot. And this is where it gets increasingly difficult to put all those observations together into one simple model. So quickly summarizing this first problem. Astronomical number of biochemical microstates, thousands of positive and negative feedback loops. All of this is taking place in a thermal bath. Energy is on the order of a few kT, 10 trillion cells in the body. And yet, small number of cell types. Cell types tend not to change suddenly. So a cell on the tip of your nose, for the most part, will stay a cell on the tip of your nose. It does not normally turn into another cell type. We also know that development is remarkably reliable. You all started out with one cell, and you ended up being the 10 trillion cells you are now. And that happened rather reliably. And then, and yet again, cells are not irreversibly locked into a particular cell type. So if needed during wound healing or differentiation, cells are plastic and can switch from one type to another. So if you scratch yourself, then the cells in your skin who once did not crawl around and divide and so forth will now start crawling around and dividing and repairing that wound. And so you're dealing with a system that has all these, at first glance, contradictory properties. And so it's largely unclear how this all works. And probably some type of statistical mechanics approach is probably needed. On to our second problem. And this is something that certainly has fascinated me since I first saw this under a microscope. And this is the classic problem of robust development of an animal from one cell. And so here is a frog embryo. Very early on, we're talking 30, 40, 50 cells. Here, a day or two later, we're talking, or this is probably 3 or 4 days later, we're talking about hundreds to thousands of cells. And of course, rather rapidly, you end up with an entire frog. And so then the question arises, how does this actually happen? What this involves is the coordinated, error-correcting, self-replication, self-organization of trillions of cells. And where biology is right now, developmental biology, is we have relatively good stories about select small aspects of this overall process, but we only have a very poor understanding of organism-wide coordination and regulation of the self-assembly process. And the movie, of course, is absolutely beautiful. This is one of the early moments of frog development. You see cells invaginating. For the first time, the system now has a clear outside and inside, and you see the spinal cord develop, and the head is about to develop, and the eyes are going to develop, and all these other things. So if you look carefully at this movie, what you see that this is not strictly a biochemical process. This is a process where cells are crawling. They are distorting themselves. They are pulling on their neighbors. There are flows of cells from the outside inward and so forth. And so this is an inherently mechanochemical process. This is not only a biochemical process. So somehow this going from one cell to 10 trillion cells, or however many it is in a frog, it's going to be right order of magnitude, involves some sort of coordinating principles that somehow all these cells are figuring out how to reliably form this structure. We're rather good at breaking this process. And here's an example from Drosophila biology, fruit fly biology. And so it has been known for quite some time that some of the basic pieces of the fruit fly, things like eyes, in some sense are modular, if you want to think about it that way. And what this means is that I can express a small number of genes at the wrong places at the wrong times, and instead of the fruit fly now having some sort of sensory device sticking out of here, it now has a second eye. And so if you look closely at this eye, it looks very similar to the normal eye right next to it. But of course now you have a fly with two eyes rather than just one. It's also very interesting to note that when you play these sort of games where you start growing wings at the wrong places or eyes at the wrong places or swapping those body parts, that some of the internal wiring is actually preserved. So as this second eye forms, it's actually beginning to make connections to parts of the nervous system and so forth. And so that is the level of understanding right now, that sometimes it's possible to break this developmental process and generate bits and pieces at the wrong places with the wrong dimensions. But in terms of having a sort of general view of how all this works that is testable and allows us to construct new things in a planned, coordinated manner, that is something we don't have yet. Problem number 3, and this is maybe one way of beginning to integrate some of these ideas, is somehow connecting genes with mechanical forces. If you look at a tissue under the microscope, what you see is not what you see if you looked into a bucket of enzymes. Instead, what you see, of course, is a highly organized object. The blue things are nuclei, green is the cytoskeleton, and red is the membrane, the cell membrane. And so what you'll see here, for example, is that the cells, neighboring cells, are actually making mechanical connections from one cell to another. And so if I were to push on this one cell, all these other cells would actually feel the deformation of this cell right here. And so a tissue is actually a mechanically coupled object. If you want to think about it like a membrane on a balloon, if I pull on that membrane, then all these cells in the environment will feel that perturbation. And so you might say, okay, fine, is this actually meaningful? And sure, if I pull on my skin, I don't pull that patch off. There are certain diseases where you will, in fact, pull the patch of skin off. But in general, that's not true. And what are the biological repercussions of mechanics being important, of mechanical signals being able to propagate through connected networks? Here's a diagram of the kind of elastic moduli that are present in your body. And so bone, as you might imagine, has the largest elastic modulus. It's very stiff, hard. Plastic and glass is way up here. And blood, mucus, and so forth has very, very low stiffness, although that's sort of not so well defined for things like fluids. But what about, say, brain? What is the consistency of your brain? It's something like Philadelphia cream cheese. So this is a rather soft, smooth material. And then epithelial cells, fibroblasts somewhere in the middle, skeletal muscle is increasingly stiff, and then, of course, anything related to bone is quite stiff. There's a good review, Nature Cancer Reviews 2009. And what you now find is if I provide different compliant substrates and I put cells from different organs on those different substrates, for example, a neuron will grow on a very soft substrate but will not grow on harder substrates, whereas various cells derived from bone need a harder substrate to grow. And so different cell types have actually different mechanical preferences for what kind of substrates they're growing on. This may begin to give you some ideas about how sort of overall organism-wide coordination of biochemistry may also have been done, because our different tissues have inherently different compliances. And so maybe compliance is one of the signals cells are using to figure out where they are in the overall body. This is zooming into this mechanobiology problem on a nanoscale. This is a membrane. This is part of the cytoskeleton. And it's now increasingly clear that the cytoskeleton is connected to the extracellular matrix through little staples. And as the cytoskeleton pulls, then certain cryptic binding sites, for example, in these staples are exposed and signaling is turned on or turned off and so forth. By the way, what's a ranked list of things that you're composed of? So number 1 is water, no question. Number 2 is actually the extracellular matrix. And number 3 are cells. So there'd still be a lot left of you if I suddenly removed all those 10 trillion cells from you. In fact, all that extracellular matrix, the collagen, elastin, bone, and so forth would still be present. And cells are continuously sensing their mechanical environment, and they're making decisions based on what mechanical environment they're in. Here is, again, one illustration of something that's beginning to be called tensional homeostasis. So the idea is that cells measure forces and compliances. Cells generate forces. We all know they can crawl and do things like that. And cells can also change their compliance, and they can change the compliance of the extracellular matrix. And they can do that, for example, by releasing enzymes that cross-link the extracellular matrix. So you have now emerging this complicated picture of where cells are sensing mechanical cues. They're modifying their own mechanics. They're modifying tissue mechanics. And all of these things are somehow connected. And so what we're dealing with is a tension sensing and actuation loop with various kind of feedback with a range of set points. And so now it becomes interesting in a cancer context. It turns out that most cancer cells have altered tensional homeostasis. And so if I take a normal cell and a cancer cell and I put them on the same compliant substrate, what cancer cells typically do is they stress the substrate more. So you can think about this as if I give you a block of Jell-O and you grab onto it, the normal cell will radially pull a little bit, but a cancer cell typically pulls a lot harder. And so this is what you're seeing in this heat map, where the stresses generated by the cells, by the cell edge, have been mapped, and they're denoted by various colors. So there's something going on in cancer that dysregulates this tensional homeostasis. And so the general problem here is to somehow start generating equations that look like this. And this is an absolutely beautiful example of probably what a lot of biology will be doing in the next 10, 20, 30 years. And so what's shown here is the Drosophila eye. This is normal. This is a mutant eye. And again, this is some variant. And you can see it has a slightly different type of packing. And the mutation that has been made here is a gene mutation. So this is a biochemical mutation. And specifically, the mutation that was made affects the degree to which adjacent cells stick to one another. So if you're thinking about sticky balloons, what you're tuning is the parameter that controls to which degree these balloons are sticking. If you look at the shapes generated by those gene mutations, they are remarkably reminiscent of what soap bubbles do. And so here are 4 cells in a modified packing, and here is the equivalent soap bubble. And of course, what controls the organization of the soap bubble, there are no genes in here. There's no proteins, no biochemistry. This is driven by things like surface tension. And what this plot is a map of concentration of protein 1 versus concentration of protein 2. And at different ratios of those concentrations, you get different organizations of cells. So you can get this cross pattern here at this combination of cadherins, this other pattern at this particular ratio of cadherins, and so forth. And so what you're doing here is you're scanning through cell organization space by tuning concentrations of molecules that control stickiness of the cells. And the equation that brings all those things together is basically a statement about the total energy of the system, somewhat loosely defined. Here we have a term that is all familiar to you, which is basically a spring term. So we have a displacement squared term plus some coefficients. And so this term is quantifying the degree to which, as I push on a membrane, it resists my attempts to deform it. Whereas these terms are not physics terms at all. These are biochemical terms. And you see here gamma sub n, gamma sub e. And so these terms make statements about the densities of those sticky proteins on the surface. And those densities are, of course, controlled by all those control networks we talked about earlier, and things like mutations and drugs and so forth. And so what you have here is an equation that now bridges all of sort of conventional cell biology and biochemistry, and is beginning to sprinkle the appropriate physics into this problem. And it's already clear, just by looking at a picture like this, that what defines the overall organization of these cells relative to one another is not only the genes. You need to sprinkle in the right physics, in this case minimization of area, to get these patterns to form properly. So what's interesting about mechanobiology is that mechanical forces can propagate along many different length scales. And so if I push on a piece of tissue, and you can all do this very easily, of course what you're doing is you're affecting the overall tissue, but that mechanical signal is being propagated all the way to individual proteins in membranes of individual cells. So what you have a very interesting possibility there for really coordinating overall activities of tissues as they form or repair through these mechanical cues. And so at this point, I've summarized 3 problems that I think, certainly for the younger crowd in this room, that potentially will be really defining problems if they decide to go into biology. And so again, that's this problem of an enormous number of possible microscopic states of the system, a relatively small number of cell types, a few hundred, and the possibility of one cell type switching to another cell type, and this is something called plasticity. It's very important in developmental biology, regenerative medicine, and wound healing and cancer. Problem number 2 is this age-old problem of going from one cell to 10 trillion cells in a self-organized and reliable manner. Again, something we don't understand very well. The final problem is moving away from a almost exclusive focus on biochemistry to mixing proteins, genes with mechanical forces that are acting on levels of individual cells and tissues and so forth. And so if you now leave the lecture hall later today, please really keep in mind that biology is about chemistry, but also mechanics. And this is something that's very easy to overlook. In terms of a specific illustration of some of these themes, I thought I'd also give you a short vignette of some actual research we do in my lab. And one thing I'd like to focus on is general interest in this biological form and function. And so what I care about specifically is how does the three-dimensional organization of biological parts relate to aggregate biological function. And a very simple example is everyone who's ever built something out of individual pieces knows, of course, that what's important for aggregate function is the pieces I use and how I arrange them, how I put them together. And this is a project in collaboration with Eric Betzik at HHMI and Hari Shroff, who's now at NIH, and Ned Wingreen in the physics department at Princeton, as well as 2 of my graduate students, Anne and Derek. And this is a project that exists in the context of something that's now being called spatial cell biology. Again, this idea of parts and relative organization determining aggregate function. And so if you believe that this is true, then one of the basic technologies you'd like to possess is reliable counting of those parts plus measurement of relative organization. And many of you in this room it will be very obvious to you why that's important. So here are some questions we could answer if we had that technology. Does this cell have 37 polymerases? Does this cell have particular numbers of certain proteins? How are those proteins organized in space relative to one another? So at this point, if you've paid close attention to your biochemistry class or cell biology class, the first question that comes to your mind is, who on earth could care? So given everything I've said so far, why should you actually care? What possible utility could there be of precision counting in biology? So I think there's 3 or 4 good reasons for that. So one of them is, imagine something is very rare. And I want to know, do you have my car keys or do you have my car keys? This is a question I can get at. If in the limit of 0 or 1 concentrations, I need to be able to count or detect as precisely as possible. The other context is fluxes. Just imagine I care about things being produced. And if I were able to look at a factory and see things being produced in increments of 1 unit, and then I can make statements about how fast things are being produced. Precision counting is also essential for thermodynamics. Remember your intro chemistry class. What's the free energy difference between 2 states of a system? Well, if you count in an equilibrated system how many molecules are in state A versus how many molecules are in state B, all you have to do to get the free energy difference is determine the ratio, right? The other thing is the precision counting, I think, is essential for robust model testing. And I'll illustrate that in a moment. So normally I talk to biology audiences, hence spending a few slides on numbers and positions. Why is that special? And I'll just focus on one example, and that is the exponentially distributed sizes of raindrops. And so I hope you've done this. I hope you've looked at the size distribution of raindrops. And what you'll find is that they're exponentially distributed, and that is related to the process by which raindrops form and grow. And then, of course, at some point, gravity intervenes, and they come falling down to Earth. But that's a detail. And so the point is, by performing this kind of measurement of the distribution of sizes, you can make statements about the basic mechanisms by which the raindrops or clusters form. And this is equally true in the universe or in any other system where there are many components that can organize themselves into clusters or drops or something like that. So the specific system we study in this case is the E. coli chemotaxis network. And so E. coli are able to chemotax towards or away from particular chemicals, things like sugars or other materials they try to eat or run away from. And that chemotaxis process is regulated by a number of chemoreceptors in the membrane, and the information is processed biochemically and ultimately transduced to a little rotary motor that turns either clockwise or counterclockwise, and that controls the statistical search that the cell is performing. And one of the key questions in the field is, how are those chemoreceptors actually organized? And it turns out that they are organized, and of course you can invoke active or passive mechanisms for such organization. Whenever you see organization in biology, you can invoke active mechanisms. Oh, there's a blueprint, and there's a molecular machine, and the molecular machine grabs and then creates little flower-shaped patterns. Or you can invoke a purely passive mechanism, where typically you're relying on say self-assembly, you're relying on some interplay of rate coefficients and things like that for patterns to form. And here's an enumeration of what we do know about the spatial organization of these receptors. This is a single E. coli, and the receptors have been labeled in black in this case. And you see that the receptors typically hang out near the ends of the cell. And so the question arises, how do these receptors get to the ends of E. coli? You can look at that with fluorescence microscopy, or you can look at this with things like cryo-EM tomography, where you get exquisite spatial resolution, but you have very little biochemical specificity. You see density, but you don't necessarily know what that density is. Here's an enumeration of some of the questions for E. coli. And I'll just summarize this by saying that most of these we can get at simply by looking and counting. Here's the traditional problem, though. If you look through a microscope and you look at an E. coli, typically the E. coli looks like a little blob. And for at least, certainly my generation, we grew up appreciating that light microscopy is limited by diffraction, right? There's certain limits to how well we can image things. And the key problem is that if I start with a surface that has molecular point sources on it, and then if I convolve these signals by diffraction, I end up with something like this. But this is a one-way function, because if I give you this image, there are an infinite number of ways of generating molecular distributions of these emitters to give you this aggregate intensity pattern. So it's a one-way function. And this is what the microscope does for us in a highly parallel analog manner. This is what we measure, and this is really what we'd like to know, though. And this was a largely unsolved problem until about 2006. And so the key insight here is that if every emitter has a unique property, and I know what it is, then I can pick it out against a background, and I can localize its centroid with a precision that's limited only by the number of photons I collect. And every astrophysicist or cosmologist, again, will find this completely obvious. And so here's the trick. The trick is to make sure that all your proteins are initially off. They're not emitting. So if you look at the sample, it's completely dark, just like a completely dark night sky. Now what you do is you stochastically activate a few of those proteins with a flash of UV light. You turn on a few, 3, 4, 5, 6. Those, by chance and on average, will be well separated. And so now I know if I collect photons, I know which molecule they came from. I can now localize its centroid with nanometer precision. And now I hit them with a big pulse of light, and I bleach them all. I bleach all the ones that were turned on. And I now do this cycle many, many, many times. So I keep stochastically activating sparse subsets of fluorophores. I localize them, and then I bleach them. And I do this thousands or tens of thousands of times. And so that gives me this technology now called POM, or STORM, or VPOM, or IPOM. The field is still trying to converge on an acronym. And I'm sure you can imagine where those discussions come from. 3 groups simultaneously described this method in 2006. And so every acronym has equally deserving proponents. This is our test system again. We have our E. coli bacterium. We have various proteins floating around that process this information, the chemical information arriving at the membrane, transduce it to the flagellum. We labeled several different components of this signal transduction mechanism, and here are the data. This is what the experiment would look like, or does look like, in normal differential interference contrast microscopy. You see the individual bacteria on a piece of glass. This is what the image looks like in conventional fluorescence. And so again, you see the receptors localized at the poles. This is what a single POM acquisition image looks like. And you see we've stochastically activated 1, 2, 3, 7 of them. So now we localize each one. This is what the POM data set looks like. And if this were a normal data set, there would be no point in zooming in, correct? All you would see are the pixels of your CCD. And you know this very well if you've ever used a CCD to connect it to a telescope. And then at some point, you just start looking at pixels. But because this is a POM data set, we can actually zoom in. And so we can now go from millimeters all the way to individual proteins, 2 nanometers. So we can now fly over this one E. coli bacterium, and each dot is one protein, and color corresponds to height over the cover slip. So now, instead of asking sort of very general questions about average concentrations of certain proteins, we can now ask questions about how those proteins are organized on a cell-by-cell basis. we can see exactly where those proteins are with respect to other parts of the cell. And we can also ask questions about how those protein distributions change as, for example, bacteria divide. And if you look closely, there's one bacterium that was fixed. Maybe it's this one or this one. It's hard to tell on the screen. It's actually this one. This was fixed just when it was starting to divide and so forth. So now suddenly you have an extremely rich data set making statements about protein concentrations and localizations. We now have to quantify these clusters. Unfortunately, we didn't have to be innovative at all, because the astrophysics community, of course, has developed very good ways of quantifying clusters. And our data set looks exactly like something coming out of the Hubble. We have a dark background and point sources. And so we can use exactly the same algorithms and approaches to investigate these data sets. So we looked at 1.1 million proteins. In your world, this corresponds to looking at 1.1 million individual stars. And now we ask how those stars or proteins are clustered and how those cluster sizes are distributed. So what's your guess? Cluster sizes are how distributed? Exponential, of course. It had to be an exponential. So here we go. Here's cluster size on this axis. This is number of clusters on this axis. And here's our red line, which is a nice single exponential. And what this immediately tells us is that asking a simple question like, what is the mean cluster size in E. coli, is actually not very well defined because the cluster sizes are exponentially distributed. Parenthetically, the red line is a particular stochastic cluster nucleation model generated by Ned Wingreen. And the basic idea here is, in terms of a generating mechanism for this pattern, is we start with a two-dimensional surface, the cell membrane. We sprinkle in proteins. Those receptors diffuse randomly. And now they can do one of two things. They can either join existing large clusters or make new clusters of their own. And it turns out that the larger an existing cluster is, the better it is at absorbing new proteins. And what does that immediately tell you? Well, the highest density of single receptors is going to be furthest away from all existing clusters. And what does that immediately give you? It gives you a very simple mechanism for generating periodic patterns. Why? If I have a large cluster at this end, where will the next big cluster be? Furthest away. So it's going to be at the other pole. If I have a cluster here and a cluster here, where's the next cluster going to be? It's going to be in the middle, and so forth. And so this immediately, at least theoretically, gives you a mechanism for generating periodic patterns based only on two or three simple things, like the rate at which the membrane is expanding and the flux of proteins in and the degree to which the proteins can diffuse laterally. Here's a concrete illustration of the benefits of being able to count. This is a cluster size probability on this axis, and I've taken our data set and I've downsampled it by the diffraction limit. And so I'd end up with a histogram with 2 or 3 bars. And you might imagine I can now fit a very large number of different models to 2 or 3 bars of a histogram, right? That gives me a lot of latitude and a lot of really interesting scientific debate about which model is correct. But you can also imagine that if I don't downsample my data, and now I'm forced with the task of fitting this distribution right here. And that is something that, again, Ned Wingreen's model, this red line, does very, very well with 0 free parameters. So this is a situation where one can be, I think, a little bit more confident in that the models we're using to describe how the patterns form are actually have some likelihood of actually being correct. And so to wind down, I'm going to do 2 things. The first thing is I'm going to spend 4 slides on something that is interesting, but almost guaranteed to be wrong. But it's so interesting that it's still worth talking about. So here's the physical picture. So imagine you're a particle or astrophysicist, and I give you a finite number of identical detectors, let's say 1,000 cameras. I also tell you that these detectors cooperate linearly. So if you snap n of them together, you get n times the sensitivity, but 1 over n times the range. And I now tell you that your goal is to build a detector network with high sensitivity and wide dynamic range. the question arises, what network topology do you use? Let's go through the different scenarios. This is chemical concentration on this axis, and this is the percent of receptors that are active. Imagine I take my finite resource, I collect it into one enormous cluster. It's now going to be very cooperative, but what's its dynamic range? It's terrible, because as soon as there are a few molecules, it's all on. And so I have an exquisitely sensitive network, but it has a relatively narrow dynamic range. And this is not a step function here because there's a lot of biochemistry that we've thrown into this model that I'm not going into now. So large clusters only. What's the other extreme? We don't cluster at all. What's going to happen? We're going to have a system that has wider dynamic range. In fact, were it not for the biochemistry, its dynamic range would be 1,000. It could be 0, 1, 2, 3, 4, all the way to 1,000 receptors on. But its sensitivity would be comparatively low. And so what if I took my finite resource and I organized this in an exponential manner, right? And this is what the red line is. And so what you see here, and this is a possibility of many, is that what E. coli is doing, it's organizing its receptors in an exponential manner to have sensitivity early on, but then trade off some of that sensitivity for a wide gain of dynamic range at higher concentrations of whatever it's trying to detect. And so if you were trying to build a detector network or something like that, any undergraduate would have come up with a very similar network topology given those initial constraints we started with. And one final thing for today is, so far all I've talked about is we've looked at the cluster size distribution, but what about relative spatial arrangement? What about this business of the clusters being at the poles and things like that? And we've already begun to talk about that. And what we find as we analyze how those clusters are relatively organized is indeed that new clusters nucleate furthest from the old clusters. And here are specific model predictions. If I have only one large polar cluster, where are the cluster nurseries going to be? They're going to be furthest away. And so this is plotting the number of small clusters of nascent clusters versus cell length. And if I have big clusters on both ends, then the cluster nurseries are going to be in the middle of the cell. And of course, if you're a fan of Alan Turing, you will say this is exactly what Alan Turing pointed out in 1952 when he was thinking about chemical morphogenesis. So this question is, how do you take a small number of components, and how do you, from that small number of things, generate a wide zoo of different topologies? And all these patterns are sensitive to concentrations of the materials, the rate at which they're produced, the rate at which they decay, and the rate at which the membrane expands laterally. And as you might now imagine, I'm a proponent of this purely passive mechanism for how chemotaxis receptors cluster. Because at the end of the day, it has to be true that in biology, you cannot have turtles all the way down. I cannot have a complicated machinery organizing another complicated machinery, organizing another complicated regulatory machinery. I need, on some level, I need to end up with particular patterns, particular structures that self-organize robustly in a way that can be controlled by things like the state of the cell and so forth. And so I like this purely passive mechanism for generating this particular spatial organization. This is stuff we haven't talked about, and so I'll quickly conclude with where we started out today was I briefed you on where a lot of biology started. And it actually started at the intersection of physics and chemistry and geometry. In the 1950s, 60s, we have this explosion of the gene, the genome. We spent 55 years sequencing the human genome. We were done in 2004. And the point of the talk was today was to communicate to you that despite all the terabytes of information we have now, all the gene sequences and all the proteomics that are coming out and all these things, the same problems that people were grappling with 100 years ago are more or less still appropriate today. And so I like to think of biology now turning into, now beginning finally to get at the real basics for how things organize and function in biology. And so I'd like to acknowledge 2 graduate students, Anne McAvoy and Derek Greenfield especially, and a variety of sponsors, and our theory collaborator, Ned Wingreen, Gavin Crooks. It's been a pleasure. Thank you.
Yes, there is.
Could you go back one, please? Yeah, so problem 1. You make the point that there's a large number of genes. I think you said 10 to the 12, 13.
Well, hang on, 23,000 genes.
Oh, 23,000. Okay.
23,000.
And the number of cell types is much smaller than that. But if you refresh your brain and look at one of your images of cells, none of those cells look alike.
Mm-hmm.
And certainly there are, the connective tissue in each cell is quite different. It's under different stresses,
mm-hmm.
and it's probably conformally different as a result. And so I guess what I'm trying to say is that in a sense, there's 10 trillion different cells in your body. They're all one of a kind. Now, I know that there's some statistical properties that make them, you can type them, right? But I think there's also this extra level of freedom that you haven't talked about in just how they display themselves in an actual environment.
Okay, well, yeah, so if you really drill down to an atom-by-atom description of every cell is going to be different, no question. Protein numbers vary and the cytoskeleton varies, and a lot of the cytoskeletal organization, the particular shapes of how the filaments are organized and so forth, that's going to differ wildly from cell to cell. And so the point I was making had to do with a coarse graining of the system in terms of sort of typical behaviors of certain cells. And even if I take, and so this all involves taking my glasses off. If I take my glasses off and look at the cells in your brain, they all have some relatively common, well-conserved features. There's electrical activity and all these things. And so that statement of a few hundred cell types is, of course, I'm coarse graining quite dramatically the system. But I'm coarse graining in a way that emphasizes typical biological functions. And so what you're emphasizing are the details of construction. And I think, and that's in fact one of the problems of bridging those two things. How can I get all those objects which have very similar aggregate functions, but can have quite dissimilar specific molecular arrangements?
Clearly, you get conclusions from large amounts of data in this field.
Mm-hmm.
Can you say a few words about maybe some novel data analysis techniques that have been developed for automated processing, and maybe some things that might even be used for looking for intelligent signals in noisy data?
Mm-hmm. So that's probably one of the areas I'm least qualified. When it comes to algorithms to analyze data, typically what we like to do is we like to just go find the person who knows most about that subject and have them help us with it. So maybe it's you, maybe there are other people in the room who could actually help us much more than I can help them. One thing that is something that we're grappling with day to day is the sheer immense volume of data that's coming out of things like genome projects, proteomics, and it becomes very quickly extremely difficult to weed through that enormous volume of information. And what I think we need are really fundamentally different and new tools to allow us to somehow explore those data sets and find patterns. And that's going to be one of the area where there's going to be a lot of breakthroughs in biology is really being able to come to terms with this massive ocean of data. And a lot of it now is sitting on hard drives. And Of course, the hard drives are doubling every year or so, but we're really almost drowning in the information that's arriving. And our ability to distill that into statements of how biology is working is something I'm certainly really grappling with. And this is going to be...
You actually have two problems. It's not only the size of the data, but it's the complexity. Your data is very much more complex than, say, astronomical data.
Mm-hmm. That's certainly true. So information is encoded in many different codes in the data, whether it's DNA. There's a spatial component to this. There's biochemistry that goes into this. And so you're dealing with very large volumes of dissimilar data. And there's also very practical problems that go into this. So what happens a lot now in biology is that one person uses their microscope to look at one sample that was prepared in slightly different conditions, different temperature, different hormone levels. Then I take supposedly the same cells in slightly different conditions with a slightly different type of microscopy. And then it turns out to be virtually impossible to then find strong correlations in the data sets when I merge them. And so maybe what's going to happen in biology is we're going to emphasize more and more multimodal data collection strategies where I have one thing pointing at one sample and I'm monitoring that sample on many different channels. So the astrophysics community has decades of a head start because, for example, in physics or astrophysics, people have been studying the sun for a long time. And it's not that different universities are studying sort of different versions of the same sun. We can all look at the sun. And typically, when you start analyzing celestial objects, you use cameras with different wavelength sensitivities. You monitor infrared and radio frequencies and so forth. And so this idea of correlating complementary data streams from one object is something that still has to catch on in biology.
So this idea of the detector, optimizing the detector topology, and so as the way we've studied it, there's a highly diverse cells, and some of them are on one end of the spectrum and some on the other. And the idea of going towards the neutral and polarity, meaning that there's a balance in the system, and then the other thing, if you can come on, is the regulatory environment, the electrical regulatory environment. And how that might, the controlling mechanism to do the polarity difference in the cell to create the diversity of the measurement to detect whatever you need to detect is that is generally thought to come from the central or the brain, and that's how your cells, how you become what you become. So I'm curious, how do you look at the electrical functional signaling at all? Or are you just purely biochemical?
So there's no electricity here.
Well, there clearly is. That's what the nervous system does.
Well,
but you're not studying it. I understand that, but.
So I'm also not a neurobiologist.
Okay.
I certainly agree that in terms of big challenges in biology, the brain is one of those. And if I were in kindergarten now, certainly trying to figure out the brain would be one of the things I could imagine thinking of as I got older. But I've decided to focus more on these things. But the brain is out of my depth.
The idea of the electrical spectrum is part of it, and this idea of polarization and the strive towards neutrality is we want to keep it diverse. So.
Maybe we can. Maybe that's a conversation best done over alcohol.
Clearly, it's the idea of figuring this out, and what we don't know is what I'm trying to figure this out about.
So you mentioned the experiment where one type of cell, the fibroblast, was converted into a neural cell. Are there any experiments which have been able to reverse that process, and what applications would that have to cancer, et cetera?
Well, the general context is this property of cells called cell plasticity. And so I can take a cancer cell, and cancer cell defined as being a cell that has various mutations in its genome and crawls and divides inappropriately and so forth. So it looks and talks and is a cancer cell. And I can apply a variety of external signals, things like antibodies or mechanical cues and so forth. and I can revert it to a less malignant phenotype. And that's just one example of cellular plasticity. And so what this is telling you is that even if you have various mutations in your genome, that doesn't force the cell to be a cancer cell. The decision of the cell to be a cancer cell arises out of the interplay of its genome and various cues it's getting from its environment, the extracellular matrix, other cells, and these things. So, and if we were able to understand that better, that now gives you a lot of control opportunities for either taking one cell type and making sure it doesn't switch to another cell type, or taking cells that have already changed into some other type and reverting them. And in fact, one of the active areas of research in our Physical Sciences Oncology Center is trying to understand the basics for reversion in a cancer context. But again, this is a much larger theme that touches on all of regenerative medicine, a lot of developmental biology, and a lot of disease biology and cancer biology specifically. And so what the field is only now beginning to learn are what kind of signal inputs provided externally to the cell force the cell to switch from one state to another. There are also, the cell itself has a variety of mechanisms to prevent those switches from happening. And that makes a lot of biological sense. You don't want cells in your skin suddenly turning into various other cell types. And so there's a lot of very basic biology left to learn. But there are enormous implications for regenerative medicine and anything related to health.
Jan, you touched on statistical mechanics and thermodynamics in your talk. In your abstract, you mentioned there might be a role for quantum mechanics that we're starting to touch now. Can you give us a taste of what might be coming?
Okay, so quantum mechanics and biology. And I approach the intersection of quantum mechanics and biology with extreme trepidation. And the general context here is, of course, that on some level, quantum mechanics is, of course, important in biology, just as it's important in everything. But typically when people ask this question, they start thinking about the intersection of quantum mechanics and the brain or various other things like that. And of course, it's important to realize that quantum mechanics applies only typically to extremely small systems and systems that are at very, very, very low temperatures. And that is a general good thing to keep in mind. It is beginning to be clear that there are certain cases in biology where quantum mechanics is important in non-obvious, interesting ways. And probably the two most informative papers along those lines have been papers published by Graham Fleming from UC Berkeley. And he studies photosynthesis. And so he's interested in a specific problem of how the photosynthetic machinery can be so efficient. The photosynthetic machinery somehow operates at room temperature, and somehow it's able to very efficiently funnel energy from photons to one site to another. And he's a spectroscopist, and what his results seem to be showing is that some parts of this energy funneling process exploit quantum coherence. And this is a very, very interesting notion that's hotly debated. And if it is true, then that means we need to think. There are features of how proteins work, despite them being at room temperature, that we don't fully understand. Does this mean I think there are some things of quantum mechanics that are wrong? Absolutely not. It just means that there are certain things about how certain proteins work that we don't fully understand. But that's certainly the most concrete example. And so there's a very nice paper in either Science or Nature describing possible use of quantum coherence to efficiently funnel energy from one part of a protein to another in a photosynthetic context.
If there are no more questions, we have a special SETI pin.
Oh, wonderful.
To commemorate your visit, and maybe it might look good under your new techniques.
Oh, splendid, yes. Okay, I will pin this on immediately. Very good.